Hello, I'm Carl Hansen, founding CEO of Abcl. Abcl is a technology company that works at the interface of computation, engineering, and biology to help improve the process of antibody drug discovery. While we work within the biotech sector, we don't consider ourselves a biotech. Biotech companies are companies that are working on developing their own drugs through to the clinic. We're also not a tools provider. We don't sell instruments and devices. Rather, we invest internally on teams, technology, and facilities to help the entire industry move their programs more quickly. Specifically, we have spent the last 8 years building a full-stack AI-powered antibody discovery engine. And we're bringing this to the industry with a centralized business model through which we partner with firms, both large pharma and small biotech companies who have needs of discovery. We take their programs internally and we deliver back to them the composition of matter for development into a drug, as well as hyperscale data sets to help them to better select their leads and to identify which molecules are going to be most successful in moving forward into a therapeutic. As a recent example of the impact of this technology and our business model is our success with COVID-19. Earlier this year, on March 1st, after having invested heavily in technologies for screening human immune responses and for pandemic response, we were able to go from a single blood sample with our partners to a first-in-human testing of a therapeutic antibody to fight COVID-19 with only 3 months. That antibody, bamlanivimab, has recently been granted emergency use authorization in Canada and the US in the fight against the pandemic. Our vision is to repeat this model and amplify this impact by working on programs across all of disease and all of biotech. Over the past 6 years, we have either worked on or signed up to do 94 different antibody discovery programs. And we will continue to invest in teams and technology to amplify the scale of our effort. And ultimately, we aim to become the de facto solution for antibody discovery across the industry. The big picture that Abcl set out to address is that everyone understands drug development could and should be done better. Drug development fails too often, it takes too long, and costs too much. What's interesting is that in other sectors, advances in technology have increased productivity and allowed us to do more. One has to ask, why has this not happened in drug discovery? We believe that one of the big reasons is a laser focus on product development at the expense of the investments that would be required to help improve the process of developing drugs. More specifically, we believe that there are 2 technology problems that stand in the way of bringing new drugs more quickly to patients. The first is limited technology, or a continued reliance on discovery technologies that have their origins in the 70s and the 80s. Specifically, if you were to go into a large pharmaceutical company and look at the discovery engine, you would find most likely that it has been built around hybridoma technology. That's a technology that was developed in 1975, the year after I was born. An immense amount has changed since 1975, and there's an opportunity to reinvent technology, to go faster, to do new things, and to do things better. The second big problem is technology access. The way that the industry works is that the large companies end up controlling and amassing most of the capabilities and modern technologies for drug development. As the industry has matured, there's been a big influx of new firms and new capital that are bringing innovation into the space, but these companies need access to technology and they typically can't get it. Abcl exists to solve these 2 problems, to push back the frontier of technology and then to democratize the industry by allowing people to benefit from those capabilities through a partnership model and centralized facilities. Specifically, we are focusing on the area of therapeutic antibodies. Therapeutic antibodies first began as a modality about 20 years ago, and starting from nothing have been the fastest growing class of new drugs over the past 2 decades. As of 2019, the global market for sales of therapeutic antibodies was approximately 143 billion, and based on traditional or historic growth rates, it would be expected that the global sale of therapeutic antibodies would reach 260 billion by 2025. We believe that that growth can actually be accelerated for 2 reasons, and one is the emergence of many new innovative firms in the industry, and second is the invention of new therapeutic modalities that either rely on or incorporate antibodies as part of their composition of matter. An example of that is shown on this slide, where CAR T therapy or cell therapy was just approved in 2019 and had about $1 billion of sales and is expected to reach 17 billion by 2025. Abcl exists to help make sure that we meet and exceed the growth potential of the industry and to participate in it, not by developing our own products, but by being a company that is helping make the process easier for everyone. From a technological perspective, we are focused on unlocking the full power of natural antibody responses. The first step to developing a new therapeutic antibody is to find a molecule or an antibody that can engage properly with the target. We believe that the best source of those antibodies is from nature or from natural antibody responses. The reason for that is that antibodies that are generated by natural immune responses benefit from 500 million years of evolution that has been essentially an arms race between all microbes on the planet and all jawed vertebrates, the result of which is a spectacular system that takes diversity, selects for antibodies that can engage with a target, and then subjects them to a rapid optimization and quality assurance mechanism, the result of which are diverse, high-quality, potent antibodies that also have the properties that make them suitable for therapeutics. The challenge today is how to better access and unlock natural antibody diversity. For that reason, we have been assembling a full technology stack that can help to generate the search space to source immune diversity, to search it, to look through the millions to billions of cells, to find those antibody-creating cells that make antibodies with the properties we want, and finally, to decode and understand those antibodies so that we can select the right ones to bring forward as therapeutics. For the past 8 years, we have been deliberately assembling a full stack solution that allows us to go from the name of a target through to delivery of a development-ready lead. The first thing to emphasize is that antibody discovery or drug development in general is not one thing. There are probably 1,000 steps that need to be done from the start of a program to handing off a development-ready lead. Along that workflow, there are several technological bottlenecks where we have looked to either acquire or develop technologies that can expand the funnel, and by integrating these together in an end-to-end solution, get a compounding return on innovation. Our technology stack begins with the name of a target. We then generate a source of immune diversity, so to define the search space for antibodies through a combination of proprietary immunization technologies, access to any species for immunization, and modern transgenic animals that mount fully human antibody responses that are tailored for the most difficult antibody discovery problems of today. Once we have generated that immune response, we then isolate the immune cells, and then we search through millions of different cells in a single day using a proprietary platform that combines microfluidic single-cell screening assays with high-throughput imaging and machine vision so that we can test the functional properties of millions of antibody-secreting cells and identify only those hundreds to thousands that have recombined their DNA to make antibodies with the properties that we're after for a given therapeutic program. Each of those antibodies can then be separately recovered and moves into a fine step where we apply automation and optimized molecular biology and genomics protocols to decode the DNA sequence that allows us to make those antibodies in the lab, and then to circle back to the larger immune systems and expand on those identified antibodies. The result being that from screening of the cells to 3 days after, we can generate hundreds to thousands of unique single-cell derived antibodies and thousands to tens of thousands of variants of those antibodies. We then select representatives from each of the antibody families, and we move them into a step that we call analyze that combines lab automation to express, purify, and characterize antibodies to generate data clouds around them that inform us on their properties in terms of potency and also developability. On top of those experimental measurements, we then layer in silico predictions, the result of which is a data cloud around each antibody that can include up to 500 different measurement points. From the beginning of our business, we recognized that antibody discovery is in fact a data science. And for that reason, we've invested heavily in the teams, the software engineering, and the data science tools to help to manage and to get information out of the data that's generated along this workflow. Moving through the platform, we have metadata on the target, the immunization methods, the species, and the tissues that are being screened. The search step generates tens of millions of images and millions of single-cell measurements of antibody properties. The find step generates hundreds of millions of raw sequence reads and thousands to tens of thousands of antibody variants. And the analyze step generates hundreds of thousands of data points characterizing those antibodies. We have built the data collection systems to organize and standardize that data, and we use that in two ways. One is to apply machine learning algorithms to circle back and improve the speed, the automation, and the precision of searching through natural immune systems. And second, to help us inform on the selection of which of the thousands to 10,000s of antibodies are best suited to move into the final step, which is an engineering step to format the antibodies for use as therapeutics. That technology step includes proprietary methods for reformatting antibodies, such as generating antibodies suitable for cell therapy or changing isotypes, as well as a proprietary technology for the generation of bispecific antibodies. This technology stack has been built up over the last 8 years through a combination of internal R&D and also through a strategy of keeping our eye on the technology landscape and looking for opportunities to bring in technologies inorganically, either through licensing or acquisition. On this slide are included 4 technologies that we have brought in from the outside. Just starting from the left here, they include transgenic animals, an in-licensing deal of the Alloy ATX-GX transgenic animal, a mouse that makes fully human antibody responses, and more recently, a $90 million acquisition of Trianni, a company that's at the forefront of using modern synthetic genetic technologies for the generation of transgenic animals that mount highly tailored immune responses that help us to solve the hardest problems in antibody drug discovery. In 2017, we acquired a company, Lineage Biosciences, and through that acquisition, Abcl now controls intellectual property at the forefront of the space of applying next-generation sequencing to analyzing the full diversity of antibodies that exist within an immune repertoire. And as mentioned on the previous slide, we use that technology in combination with our single-cell screening to expand diversity around antibody hits that have been found, and that allows us to more quickly move from good antibodies to great antibodies for development. Finally, earlier this year, we announced the acquisition through an asset purchase agreement of a protein engineering technology called OrthoMab. The OrthoMab platform is a computationally directed protein engineering solution that allows for any 2 human antibodies to be recombined into a single molecule that can simultaneously engage with 2 targets. On this slide at the bottom are some examples of great companies that are also recognizing the opportunity for innovation along various verticals in the workflow from the name of a target through to development, through to the generation of a development-ready lead. We have technologies that we believe stand toe-to-toe or have advantages at each of these different verticals, and Abcl is completely unique in having invested to deliberately build a modern solution that integrates all these technologies together into a workflow that allows you to go quickly from a target through to a development-ready product lead. Abcl is also advancing a highly unique business model to bring our technology and our capabilities to the industry. Underlying Abcl is a central thesis of technology advantage. We are investing long-term in teams, facility, and technology that can help to move the needle for drug discovery. Because we know that we're going to work on the hardest programs in the industry, we can invest aggressively in making sure that we have state-of-the-art technology and capabilities for our partners. Further, we have recognized that there is a flywheel effect in running this partnership business. Our partners come to us with some of the most challenging discovery problems, and through that work, they direct our R&D to help to improve our platform and make it better with every iteration. As we do that, we also gain know-how and we accumulate data assets that we maintain use rates on that allow us to use artificial intelligence to further increase the speed, the economy, and the capabilities of our discovery engine. We have found over the past 4 years this creates a flywheel effect where technology advantage attracts partnerships. Partnerships allow us to invest aggressively in technology and capabilities, and by running programs, our product, which is our teams, our people, and our capabilities, get better. As we begin to scale this business over the coming years, we expect that this flywheel will accelerate, and we aim to have an increasing technological advantage that helps our partners more quickly reach their goals and, of course, helps to bring new therapies more quickly to patients in need. Over the past 6 years, we have worked on or signed up to do 94 different antibody discovery programs. On this slide is an example of some of our announced partnerships. And what I want to highlight here is that our partners include some of the very best firms across the entire spectrum of the biotech industry, from the small companies that are advancing novel technologies that are based on antibodies, as well as companies developing new therapeutics based on new biological insight, right up to the large and highly enabled firms, such as the large pharmaceutical companies that come to us when they're looking for solutions that help them to expand their capabilities and to solve drug discovery problems against target classes that have traditionally proven refractory to the conventional technologies. A recent example of that is our deal with Eli Lilly that set the stage for our collaboration on fighting COVID-19 and the development of the antibody therapeutic bamlanivimab. I'd like to emphasize this particular collaboration as an example and a proof point of our business thesis and our business model. Leading up to the pandemic, Abcl had spent 8 years developing technologies to solve the toughest problems in the industry, and we had also specifically focused on accelerating our platform for pandemic response, and in particular working in a program with the Department of Defense, had put in place technologies for the rapid screening of human immune responses to find antibodies that could be used to prevent or to treat viral pandemics. Earlier this year, when the COVID-19 arrived in the United States, we deployed that platform, and on March 1st, we're able to obtain one of the first samples of a patient that had recovered from COVID-19. Over the period of 6 days, we applied our platform to screen that patient sample. That included testing the antibody properties from approximately 6 million immune cells, which led to the identification of 2,200 cells that make antibodies against the virus that were sequenced to obtain a panel of 500 unique monoclonal antibodies. Shortly thereafter, we were able to connect with our partner Eli Lilly, and working with them, we're able to bring that molecule to first-in-human testing on June 1st. And of course, that molecule has recently been granted emergency use authorization only 9 months after beginning the program. We believe that this is a terrific example of investments in technology and capabilities helping to accelerate drug development, and that the impact of those technologies is greatly amplified through partnerships with groups that bring complementary expertise. In this case with Eli Lilly, the rapid manufacturing, the clinical development, and now the commercial development. We also believe that this example underscores our main business model, which is in striking deals and contracts that allow us to participate in the success of our partners and the benefit of patients that receive the new therapies.
Thanks very much, Carl. My name is Andrew Booth. I'm the CFO of the company. I'm going to take a few minutes here to discuss the business model and the financials of the company. First of all, in the business model, I think the bamlanivimab case is an excellent example of how our investments in teams and technology have really put us in a position where we could put it to work when the world needed it the most. Quite confident that it will, and we're very optimistic that it will also be a great proof point of the business model and how we've chosen to take our technology to the market. Here you see on the slide an example of how that business model works. The overarching theme is that we share in the success of the molecules that we discover. And in fact, we have such conviction in the quality of the products and the molecules that we discover that we're willing to defer the lion's share of the economics to a point in time when the success of our customers is clear. So we get paid for what we do with our discovery research fees. As a result of that, we pay very close attention to this portfolio that we're creating as we're doing our discovery work that is long and diversified on biologics. We see that in these key metrics that we have presented in our S1. I'll walk you through each of these charts in turn. So on the left, we have the number of partners, and I'll take an opportunity to explain the difference in the shading on this graph. So in the early years, we didn't always have downstream milestones and royalties with our partnership programs. Sometimes it was because we were still cutting our teeth with the technology, or in many cases, they were programs with DARPA or the Gates Foundation where downstreams didn't really make much sense. You can see that that's leveled off in the recent years, and we've been focused on partners that are pursuing bona fide clinical therapeutic programs. You see in the center chart the number of programs we have under contract. We've been doing multi-year, multi-target deals with many of these customers certainly in the recent past, and you see how that's been accumulating and accelerating this growth of the number of programs under contract. So once we have these programs under contract, it's up to our partner then to call a target where we'll agree with the statement of work, arrange reagents, and then actually start the discovery program. And that will be the chart on the far right. Here you see the number of program starts, and this is an indication of how many drug development programs are really live out there in discovery or in preclinical work. And in the case of bamlanivimab, actually in the clinic or approved as a therapeutic. It is also a great measure of the capacity that we have within the organization in order to take on discovery programs year over year, and tracks, as you would expect, to the revenue that we recognize over the period. On the next chart here, you see how that revenue has developed over the years. This is US GAAP revenue. And when you're a CFO, this is a very comforting chart of year over year growth, very steady and actually explosive in 2020, as you can see on the chart from Q3 of last year. Of course, in the 2020 number, we do have some milestones from the Lilly molecule, but that only accounts for about 8 million of the 25 million you see here, meaning that the underlying growth of the partnership business is still 100% year over year. As you see along here, we've had very efficient use of capital throughout the period here. We've been cash flow positive from operations, and we've complemented a lot of our economics with very clever use of non-dilutive financing. Along this path here, we've also managed to make significant investments in facilities and equipment in building out the capacity within the team, and been able to afford inorganic activity, the acquisition of Lineage Biosciences in 2017, the acquisition of a bispecific platform in 2020, as well as the Trianni mouse that happened recently. I think importantly along here is also the headcount growth that we've had. Up to the end of Q3, 174 employees. This is especially important because while we have efficiently and effectively deployed capital in the past, we have built the team over the years to continue that track record of deployment of capital in a cost-effective way. And we really have the team in place in order to do that. So we're proudly founder-led with both Carl and Veronique Lecault, who's a director and chief operating officer. Rounding out the board is John Hamer from DCVC Bio, John Montalbano, who has joined us recently as a director and is the audit chair, Peter Thiel, who has joined recently as a director, and Michael Hayden, who's been a director for well over a year, an established scientist, entrepreneur, and formerly the CSO at Teva Pharmaceuticals. In the executive team, we have Esther, Kathleen, and Sherry, who have been here really since the beginning. And in fact, the lion's share of the employees are under Esther, Kathleen, and Sherry, either doing platform development or discovery partnership work. In addition, we have Adam Clark, who has doing platform development with a focus on difficult targets. Bo joined us recently from BMS, taking on translational research and helping our customers move closer to the clinic. Tiffany has been leading up communications and also our the way we present our corporate image. Veronique heading up operations, and rounding it out, both Trin and I are heading up the leadership of the more corporate functions. So really a passionate team with a lot of experience, ready to take the company to the next level. And I would happily say that everyone here is having a lot of fun as we've been continuing on this great journey, and we're looking forward to the next steps. So that is everything for us. I want to thank you for your attention, and we'll be happy to answer any questions as we move through the roadshow.